Hey folks, and welcome to another edition of Zog Science. Today we're going to be beginning our series on biotechnology, which is by far my favorite part of biology. We're going to be starting off with genetic engineering. So, what is biotechnology? Well, biotechnology is basically using any sort of manipulation of DNA or manipulation of the central dogma that we've kind of learned about in order to create a product. A lot of companies are right now involved in biotechnology, especially here in North Carolina. We have a large biotech industry. And genetic engineering is the foundation for almost all biotechnology. And genetic engineering is basically the direct manipulation of genes so that we get a particular protein produced that we want. So let's talk about DNA cloning, right? So we want to make some copies of DNA. And in order to do that, we're going to follow these general steps. Now, there are differences kind of depending on what you want to do, but these are just kind of the general steps. First off, we're going to obtain a gene of interest. So whatever it is that our piece of DNA that we want to clone, we need to actually obtain it from our source. Sometimes that needs to be modified. So, for example, if we're going to get a piece of DNA from a human and we want to put it into a bacteria, we're going to need to modify it because we need to get rid of the introns. Then we've got the DNA that is going to be combined with a plasmid. Remember, plasmids are pieces of small circular DNA. We take this new plasmid, which we call recombinant DNA because we've recombined it with something new. We're going to insert that into a bacteria. When the bacteria reproduces, it's going to begin making copies of our plasmid. Our plasmid has a gene of interest, therefore our gene of interest will also be copied. So again, those are just kind of the general steps. All right, so here we go. So we've got a picture showing us the steps of the DNA cloning. Again, remember that eukaryotic DNA has introns and exons, so it's got to be modified before being added to the bacteria. So we've got our gene of interest, right, inserted into a plasmid. Plasmid is put into a bacterial cell. Bacterial cell reproduces. We can then either take the copies of the gene, use that for different things, or we can harvest the proteins and use the proteins in different ways. So both the protein and the gene itself are useful in research. Now, how do we actually go about cutting the DNA, right? Because we've got this gene of interest that we need to cut from the original DNA, and we've got the plasmid which we need to cut. Well, we use these wonderful tools called restriction enzymes. Remember, we talked about restriction enzymes when we mentioned viruses. Bacteriophages, when they infect bacteria, they can sometimes get cut up by restriction enzymes. And they cut at a specific sequence of DNA, which is known as the restriction site. That creates restriction fragments, which have sometimes sticky ends, and then those will combine with other complementary strands. So if we take two pieces of DNA, cut both of them with the same restriction enzyme, we get the exact same complementary sticky ends. They will now stick together, forming a recombinant DNA molecule. Now, they don't always recombine exactly the way we want them to, so we have to pick our restriction enzyme carefully so that it cuts at a place that is going to actually be beneficial, be useful. And there are a lot of different examples of restriction enzymes. Over here, we can kind of see there are a wide variety of different microorganisms that they have been isolated from. The restriction enzyme itself is named here. You can see the cleavage site. So this is the specific spot where the cutting will occur. And you can see the products of that, the various sticky ends. Now you'll notice right here that this enzyme, it actually cuts in one location. And when it does that, we call those blunt ends. So those enzymes, when they cut, they actually don't form sticky ends, so they can recombine with anything else, which can be useful, right? Because now we can combine any two pieces of DNA. Doesn't necessarily have to be cut at a specific spot. A little less precise, right? A little less precise, not always going to get the complementary DNA strands lining up back together again. So the creation of the plasmid, right? We're going to take our genes of interest and the plasmid and cut them with the same restriction enzymes. So again, we get the same sticky ends. The DNA is allowed to hybridize or come back together. And it's important when we create our plasmid to include reporter genes. And the reporter genes are going to help us tell if transformation has occurred. For example, beta-lactamase, GFP, lactase. So here, we've got our P-glow plasmid. This is the plasmid that we're using in our transformation. We've got 4 genes that we made sure to include. The origin, which is where replication of the plasmid begins. Arabinose C, which is a regulator protein for the GFP. We've got GFP, which is green fluorescent protein, and we've got beta-lactamase. Beta-lactamase allows the bacteria to survive in the presence of ampicillin. So if we put ampicillin on our petri dish, we expect that only those bacteria that have resistance to the ampicillin will be able to reproduce, which is good, because then we are selecting only those bacteria that have actually been transferred. So let's kind of take a look at the reporter genes in action. So what we've got here is we've got a lac operon or a lactose gene, which is going to help kind of express whether or not lactase is present or not. And what we do is we have selected a restriction site that is in the middle of that gene. So if that gene gets cut out, right, and we add in restriction fragments, then that gene will not be expressed. Therefore, we know that recombination has occurred. So we've got different bacteria, and they're going to grow, and those of them that are disrupted, therefore they are recombinant, are going to appear white. Those that are not disrupted are going to appear blue. Again, the GFP protein is used a lot for this because it glows under UV light, so that's really good for being able to determine whether or not replication has occurred, or not replication, transformation, as well as the ampicillin resistance. That is also really good because now you know that you're only getting those bacteria that actually are new. So that's really good. All right, some other vectors. Not only bacteria are used, viruses can also be used as vectors. Vector is just something that is going to be carrying a trait for us or carrying some genes. What happens is that a lot of times these are retroviruses, so they start off with RNA. The RNA will then insert the DNA into a cell, which then can be cultured, and we can inject the engineered cells into patients. It's used a lot in gene therapies to insert a normal gene into something, but it's not really the best. It still is fairly unreliable. So this is still pretty new technology, still trying to get a handle on using viruses to do gene therapy. We also can have these really cool things called gene guns, which fire DNA-covered pellets into plants, as well as some other organisms, but usually plants. And what that does is that puts in the DNA right into the plant cells and right into the nucleus, actually, even, and that will transform it, and you have a new tissue. Now, one of the really cool things that we can do if we want to do some genetic engineering is we can create cDNA libraries. And cDNA is complementary DNA. And what happens is that we isolate mRNA found in a cell. We then add some reverse transcriptase to it, which will create DNA, right? And now we have a brand new piece of DNA that is our cDNA. And that DNA is built directly off of the mRNA, which means that that DNA will directly lead to whatever protein is found, right? So what we can do is we can take a look and see, okay, what is all of the RNA molecules that are inside this particular cell, right? We reverse back and find whatever DNA is there. Now we know all of the genes that are being expressed in the cell. And we'll talk about a little bit more in depth how that actually happens, but it's going to use the DNA chips, which we saw a little bit earlier in the year. But we're going to use DNA chips to kind of help us with that process. But this is a really cool way to be able to figure out all the DNA that is actually being expressed in a particular gene. What we can do then is we can take a disease cell and a normal cell and compare the genes that are being expressed, right? And now we're able to kind of see, okay, what genes are turned on? What genes are turned off? How is that affecting whether or not this cell is diseased or not? So pretty interesting stuff and really a cool piece of technology. Now, what are some applications of genetic engineering? Some actual real products. Well, human insulin, as well as some other compounds, including clotting factor, can be produced by either bacteria or other organisms, or in the milk of other animals. So, for example, there are goats that in their milk, they are producing spider silk. In their milk, they are producing that human clotting that I was telling you about, that factor. And then we can use those to help people out, right? So human insulin being produced by bacteria, much better than just using pig insulin, which is what they used to do. It's cheaper, and also it is much more efficient because it's actually human. We can have crops that are modified to produce additional nutrients. So in the picture here, you can kind of see golden rice and white rice. Golden rice has extra nutrients in it, so those people that might be a little poorer, right? They grow that golden rice, they're able to get all the nutrients that they need. They're not going to be malnourished. We can also have crops that are modified to resist pests. There is a protein that, when added to particular plants, will help destroy caterpillars that eat the plants. It'll help kill them, keeping them safe without having to use a whole bunch of pesticides. And then finally, we can use food animals. We can genetically engineer them so that way they will grow larger. For example, this salmon in the back is one that is genetically enhanced as compared to the salmon in the front, which is a normal one. Now, how do we actually transform those plants? Because it'd be kind of, kind of weird. You don't really think about bacteria being able to do this, but we actually have this wonderful bacteria called Agrobacterium tumefaciens. And what it does is it actually creates a tumor in cells, in plant cells. And so how it does that is it inserts its own plasmid called the TI plasmid. So what we do is we make our TI plasmid recombinant, right? And then the bacteria will actually insert its DNA, that T1 plasmid, into the cells, and now the plants have that, which is really, really cool kind of technology that allows us to be able to transform plants, not just bacteria. So really cool stuff. It kind of acts like a virus to plants. So anyways, thanks for watching. This is just kind of the start of the biotechnology, and the next one we'll look at some more really cool applications. Thanks for watching, and I'll see you next time.